Well, thanks so much. Really appreciate the invitation to chat with all of you and to have attended this conference. A lot of fantastic talks that interestingly collide a little bit peripherally with some of what I will talk about, with a focus really here on cardiology. Here are my disclosures, and towards the end, I'll hope to make it relevant for everyone throughout, but also mention our Eureka digital research platform that is really built to facilitate mobile health quite broadly. So hoping this might also help foster some collaborations. So, you know, in general, with much of the research related to studying sensors or using wearables, there's often this tension or harmony, as you choose to see it, between a cool new gadget and a research question. And sometimes there's a little too much emphasis on the cool new gadget. And ultimately, one of the things I hope to argue here and demonstrate is that having a strong foundation in traditional clinical research methods, epidemiology, biostatistics, and ultimately the relevant clinical question is by far the most important thing, and to view these sensors and technologies really as tools to help us get there. So this is a brief outline of what I'll talk about with a focus on patient-centered questions, very much in line with what we heard about from Dr. Gerson from PCORI, and then leveraging existing device data for analyses in hopefully efficient ways, and then ways to think about using those devices for your own purposes, and perhaps that may bring out the most novelty, and then finally a little bit about our Eureka digital research platform. So first, to hone in on this notion of patient-centered questions. So this actually arose from a PCORI-funded summit where we brought together patients and investigators and an interest group in atrial fibrillation, kind of organically formed, atrial fibrillation being the most common arrhythmia, common cause of stroke, dementia, death. And patients told us that they were most interested in understanding their individual triggers for discrete episodes of atrial fibrillation. And I'll note that led to us performing this survey among our worldwide internet-based healthy heart study participants, and the subset of those with AFib, where we just asked them, what do you think triggers your atrial fibrillation? And by far the two most common reported were alcohol and caffeine. And so I'll talk a little bit about both of those substances, which I think are especially interesting to study because they are so ubiquitous, and there are somewhat conflicting data regarding their actual health effects. So in terms of alcohol and atrial fibrillation, this was an important meta-analysis that brought together a lot of data on the subject, looking at those who tend to drink more alcohol and their subsequent risk of developing a diagnosis of atrial fibrillation, and finding that, in fact, there was such a relationship. Now, one of the issues with these sorts of studies is, of course, that they rely heavily on self-report of alcohol consumption, which perhaps not surprisingly is not always reliable, especially among those that tend to drink more. So we conducted this study. This was R01 funded by the NIAAA, where we took patients with paroxysmal atrial fibrillation that really drank, as long as they drank some alcohol, minimum of once a month, and fit them with ankle sensors and a continuously recording EKG monitor called a Zeo patch, some of you may be familiar with from your clinical practice. And as part of clinical practice, we often ask our patients to press the button on the Zeo patch to timestamp when they have symptoms. In this case, we told them not to do that, but rather to only press the button when they consumed a drink of alcohol. We also obtained these fingerstick blood tests for what is called phosphatidylethanol, which is kind of like a hemoglobin A1C for alcohol, reflecting alcohol consumption in the past couple of weeks. And we were able to validate those button presses by both the phosphatidylethanol or PEth results as well as the transdermal ethanol sensors, and found that indeed drinking one drink of alcohol essentially doubled the risk of a discrete atrial fibrillation event happening within just a few hours, and drinking more heightened that risk even more. Separately, just looking at the data from the transdermal alcohol sensor, we found that whether it was the peak alcohol concentration or the area under the curve, those both also predicted a discrete episode of atrial fibrillation. And we were fortunate to get a lot of press on this, including a kind of a feature in the New York Times, which I think demonstrates the potential kind of power and influence of taking a patient-centered research question and applying these novel technologies. You know, it had been long reported that patients had told us, at least our atrial fibrillation patients, alcohol is a trigger, but it had been essentially impossible to really prove that because you need to follow people continuously in real time in their ambulatory lives. And here we were able to accomplish that. And it's been very, I should say, gratifying. I get emails all the time now from patients who read that article with atrial fibrillation, said that in response, they've reduced their alcohol consumption and enjoyed, many of them at least, less AFib. Okay, moving from one of the most commonly consumed drugs in the world to the other, caffeine or coffee, where here there's kind of an opposite experience as we look at the real evidence base. The conventional wisdom is that coffee enhances palpitations, but there's growing evidence this may in fact not really be the case. This is just one example that we've published recently using data from the UK Biobank, where we showed that those who consume more coffee experienced a lower risk of instant arrhythmias. Of course, once again, this is limited by self-report, and we know that may not be reliable. So we've recently completed enrollment and initial analyses of this trial that we called CRAVE, which stands for Coffee and Real-Time Atrial and Ventricular Ectopy. We use the Eureka, our Eureka digital research platform, I will tell you about more towards the end, and fit these essentially volunteers with multiple sensors, including a continuously recording EKG device, the Zeo patch, the Dexcom, and I should call out, thanks to Tom, who just finished giving his talk, and others at Dexcom, they actually generously donated these continuously recording glucose monitors. Incidentally, we excluded those with type 1 diabetes. We fit everyone with Fitbit devices and used a API that we already have, an integration with our Eureka platform to record both step counts, and also used a Fitbit that also has been fairly well validated to measure sleep. And then we collected DNA to genotype for caffeine metabolism SNPs. Now, and then participants received instructions every day to either go ahead and consume coffee as you normally would versus abstained from coffee day to day for 14 days. And of course, a natural question then is, well, how do you know they're actually following their randomly assigned instructions? So we made use of this feature that we have now in Eureka that we've called geofencing. This was a proof of concept study where we passively monitored geolocation and looked for our healthy heart studies to trip the geofence for hospitalizations in this particular case, and validated that both for patients we knew exactly when they were coming into UCSF and then around the US for various hospitals around the US. So in CRAVE, we actually geofenced for coffee shops. Also, we told participants that we would reimburse them for all coffee consumed, whether they were supposed to or not, as long as they gave us a date-stamped receipt. And I will say that compliance with this was excellent. The data or the results are embargoed, as I'm actually presenting this as a late-breaking clinical trial at the American Heart Association in about 9 days. But I can say that there are several highly statistically significant results that I suspect will be of interest related to cardiac ectopy, which we've incidentally learned both premature atrial contractions and premature ventricular contractions are actually likely very relevant, as well as effects on physical activity and sleep. So moving on and thinking about this a little bit more and how we can really make full use of these sensors, you know, I talked about alcohol and caffeine, but of course, most of the people reported these various and often idiosyncratic triggers that they felt were important to themselves, and that was kind of the whole point of this. So the question is, well, can we somehow make use of that and really do this personal sensing and with our digital phenotyping? So this led to a PCORI-funded trial that we called iStop AFib, Individualized Studies of Triggers of Paroxysmal Atrial Fibrillation, where we worked with patients, used our Healthy Heart Study, a organization called stopafib.org, and another patient, Debbie McCall, has a large Twitter following of a AFib interest group to invite symptomatic paroxysmal AFib patients that were interested in testing their own trigger. We developed this study on using Eureka, and participants were then randomized to just track their AFib versus to undergo their own N of 1 trial. And those randomized to the N of 1 trials were presented with a menu of that list of triggers that we had ascertained from our survey, and they could pick one, or they could write one in. And then they were randomly assigned for 1 week on, 1 week off for 6 weeks. It was actually, the random assignment was actually either on off or off on, to go ahead and test your trigger this week. You told us that alcohol is a trigger for you. This is your week to test your trigger, versus, and on the off weeks, please make sure to avoid all alcohol. And they received reminders. And at the end, we assessed AFib severity and quality of life. We sent them all these AliveCore cardiomobile devices that are essentially electrodes. Oftentimes these can be embedded in a smartphone case that pairs with the microphone on smartphones to record a single lead ECG, and there's an FDA cleared algorithm to overread episodes of atrial fibrillation. We also then had a board certified cardiologist overread any strips called as positive. For those that underwent their N of 1 trials, after 6 weeks, we then gave them this sort of visualization, and we tried to make it fairly straightforward to show them in yellow, here are the weeks, and this actually matched with the actual calendar dates. Here are the dates where you were assigned to test your trigger. In gray, here are the dates you were assigned to avoid your trigger. Here's where you had AFib or not. And then we had an R program that ran in the background that was able to generate a probability, calculate a probability based on those results that they did indeed, that their trigger did or did not actually influence their atrial fibrillation. This also is going to be presented at a late breaker at the American Heart Association later this month. Okay, moving from patient-centered questions to this notion of leveraging existing device data that may be an especially efficient way, especially for those in training or junior faculty that are trying to make full use of things already available efficiently. And I'll share this experience that we had with a breathalyzer company. So there is this company called Backtrack that markets Bluetooth-enabled breathalyzers to the general public. Presumably people want to check before they're going to go out driving. And the lesson I would hope to share here is, if you're working with a company that has a Bluetooth-enabled device or a device that pairs with an app, that company has a lot of data that is just sitting there, and they're arguably incentivized to work with you, presumably a primarily or exclusively academic who is interested in science and publishing peer-reviewed papers, and that's potentially great exposure for them. So this is what we did with this company that we were working with on some other projects, realizing that they had tens of thousands of customers around the world. And I just asked them, do you actually have data on when people use your devices, what the blood alcohol concentrations are, where they were? And in fact, they did. We developed a data sharing plan and obtained institutional review board approval. And one of our faculty members, Kirsten Aschbacher, who's a data scientist and psychologist, applied a machine learning algorithm that we were able to publish in Nature Digital Medicine, demonstrating behavior patterns that would predict when the blood alcohol concentration exceeded 0.08%. And then separately, we used those data essentially as instrumental variables. This notion that, so the idea was we would identify dates where people tended to use the devices more and when the blood alcohol concentration was higher, in the hopes of identifying essentially recurrent, nationally recognized events to use as an instrumental variable to then test to see if those dates were associated with more atrial fibrillation emergency room visits using the AFib database. And indeed, we found that those events listed here on the left were more often associated with AFib visits to the ER, suggesting on a population level that alcohol consumption is really contributing meaningfully to AFib visits to the ER. A couple of points about thinking about using these devices for applications for which they weren't initially intended. So we actually published the first study to demonstrate that an Apple Watch, really any smartwatch, could detect AFib. This is before Apple had this bright idea, where we recognized that smartwatches were collecting information on heart rate, that especially in workout mode, that was somewhat variable, that AFib could reasonably be inferred from a more irregular heart rate, especially at rest, and these devices have accelerometers, so we could incorporate that as well. So we, again, trained a machine learning algorithm using ambulatory participants in our healthy heart study. We then validated that in a very well phenotyped sedentary population undergoing cardioversion, so we knew they had AFib, then we knew they had sinus rhythm, and then validated again in an ambulatory population. Incidentally, that the accuracy was much reduced in that ambulatory population, and there's a lot to discuss about screening for AFib, whether that is appropriate or not, and the potential downsides and unintended consequences of utilizing these devices. Happy to get more into that if that's of interest in the question and answer period. And then there's this idea of, if you are collecting these data anyway or have access to these data, to then leverage understanding health effects of secular trends of some event where you would expect to change. So here we leverage the fact that we had just invited our Healthy Heart Study participants to connect up various devices, such as their Fitbits. And this is during one of now, unfortunately, too many wildfire seasons. It's become kind of the norm, unfortunately, in California, where we really had to, this is pre-pandemic, almost shelter in place because the air quality was so poor, but demonstrated that there are these health effects that folks may not have been fully aware of, which is the sedentary behavior that many are kind of forced to endure when sitting inside. And then similarly, along very similar lines, we utilized data from connections with Bluetooth-enabled scales, also from our Healthy Heart Study, to demonstrate that people on average gained a lot of weight during the shutdown. This also garnered a lot of press. One of the pinnacle moments of my career was Stephen Colbert mentioning this study, but again, very gratifying to try to make sure that people understand the downsides of sedentary behavior, to think about the lack of physical activity, the perhaps greater availability of food in the setting of the lockdown. And then finally, in the last few minutes, as mentioned, I wanted to describe a little bit about our Eureka digital research platform, which again is built to facilitate really mobile health-based research very broadly, certainly not just in cardiology, not just at UCSF, but nationwide. So we view this as having three components to it. One is this flexible, scalable mobile health research program. We also have accessible e-cohorts. I've mentioned the Healthy Heart Study a couple times that has now more than 300,000 individuals that have essentially said, yep, I'm interested in contributing to digital health research. I'll tell you about another one, the COVID Citizen Science Study, that has similarly become a platform accessible for various research projects. And then we have a data repository and can perform analytics. So we can provide eligibility and consent. The great majority of these studies are remote. We have a randomization engine, although many of our studies are observational, a robust survey engine, many device and app integrations. We can collect data from phones, and then we have a messaging queue that can utilize text, push notifications, or email reminders. The idea is that it's really one app, and we can deliver multiple studies on that one app, and we can make those studies exclusive. So from the perspective of the participant, it's not that they're necessarily seeing other studies unless that's of interest. One of the benefits to that is when there's a new version required or some change on iOS or Android, we don't have to rebuild the app, create a new version. We just can work on the one full app, and then we can also populate changes and do updates quite rapidly from the back end. We also can provide branding. We have designers that are very savvy in terms of user experience. This does not need to be branded UCSF by any means, for example. We also can have variable sorts of campaigns. So we've had studies where folks are interested in identifying what site people are coming from or what sort of campaign led to recruitments. So we have customizable text backs or QR codes or URLs. We often have done many studies where we are co-enrolling with some extant cohort or medical institution, and we can provide unique participant codes to then link up data. Those are usually embedded in the link, so it's no problem or extra effort needed on the part of the participant, but we're linking back to, for example, their Framingham Heart Study data or childhood cancer survival study data. I'm doing a project now working with the American College of Cardiology, NCDR, registry, and then we've also done with this with the CoreNet institutions, for example. And we are built to be as adaptable and nimble as we can. So we have a mobile app. We also can have understanding not everybody has a smartphone. We have web-based experiences for participants as well. And participants can have both and toggle back and forth. As mentioned, we can do remote studies or in-person studies or a hybrid. And in fact, a couple of the studies I shared, our geofencing study, our Apple Watch study were both examples of hybrid, where we had conventional in-person enrollment and then remote components. As alluded to, we have many device integrations that are readily available. Several companies, blood pressure cuffs and scales, Fitbits, the AliveCore device, and then data collection from smartwatches and iPhones, for example. As I mentioned, we have this study now that this slide I made maybe 10 days ago. We're now almost to 100,000 participants. This is a mobile app-based study collecting symptom data, infection data, information regarding vaccinations and side effects from people from all over the world. This, by the way, is another kind of repository of data where we have way more data than any of us have time to get to. So if folks are interested in secondary data analyses, there's a lot of interesting things to dive into here, and happy to chat about those possibilities offline. We've also thought a lot about retention and engagement, and of course, this is always a challenge in clinical research, but especially in remote and technology-based research. So thinking about how to keep people engaged, such as via newsletters and a study blog, as shown here for our COVID citizen science study. And again, the idea here of Eureka and our charge really from the NIH is to provide something that's useful to you as an investigator to pursue your particular research interest. And again, happy to chat offline to anyone that may be interested. So in conclusion, thinking about digital phenotyping and personal sensing with a frame of cardiology, but hopefully more broadly relevant. Number one, patient-centered questions are often the best drivers. So starting there, not necessarily starting with a technology per se. Leveraging existing device data can be a very efficient way to conduct this sort of research. And then thinking about using existing device data for your own applications can be a way to introduce novelty and innovation. And then again, please consider using our Eureka digital research platform. That's all I have. Thank you so much.